Ever get curious about something that sounds pretty simple at first,
Oh,
but then you realize it's
yeah.
like this huge, complicated thing with
Mhm.
so many layers,
Yeah.
and it's actually like super important. That's kind of what we're dealing with today when we look at microbial
It
resistance
is a big one.
to antimicrobial
Yeah.
agents.
Yeah.
It's a big deal.
Huge implications for, treating infections, how we approach healthcare.
It's everywhere.
Ah,
So basically, we're going to like really try to unpack,
yeah.
paint a clear picture
Yeah.
how these tiny little organisms, these microbes,
Hmm.
can actually fight back
Yeah.
against
Right.
the drugs that we're trying to use,
They evade them. Right.
get rid of them.
Yeah.
So in this deep dive, we're going to
Yeah.
break down all the ways, all the fundamental mechanisms that this resistance
Exactly.
pops up.
It arises. Yeah.
Okay, so the sources are laying out these like different types of resistance. The first one that kind of jumps out is
Right.
natural
Yeah.
resistance.
Yes.
What's that all about?
Well, that is the fascinating thing, that some microbes are just inherently resistant.
Okay.
They're born that way.
So they're just like, nope, not going to work on me.
It's just in their genes. They never even saw the drug.
It's like built in.
It's part of who they are, right?
Well, their fundamental characteristics are just like,
Think of it like this. You know, if a door is locked to a specific key,
uh,
any key that doesn't match that lock isn't going to work. So some bacteria, they just naturally lack the lock that a particular antibiotic is designed to target.
I like that. So it's like a mismatched lock and key situation.
Exactly.
Okay. Can you give us some specific examples?
Oh, absolutely. So one really clear one is mycoplasma.
Okay.
These bacteria, they don't have a cell wall. And, penicillin, how does penicillin work?
By attacking the cell wall.
Exactly.
Look at that.
It attacks the cell wall. So if you don't have a cell wall, penicillin's not going to do anything to you.
Makes sense.
Another great example is gram-negative bacteria. These guys, they have this outer membrane.
Oh, yeah.
It's like a security screen. It prevents larger antibiotic molecules from even getting inside the cell. So something like vancomycin, big molecule, can't get through that outer membrane.
So they just like bounce right off.
Bounce right off.
Too big.
And then some bacteria, they have these innate efflux pumps.
Okay.
These are fascinating. These are like little bouncers inside the cell,
I love that.
kicking out unwanted guests. In this case, the unwanted guests are certain antibiotics.
So they're just like, you're out of here.
Not letting them in or kicking them right out.
Get out of here.
So yeah, it's not something they develop. It's just how they're built.
Right. They're just born that way.
Yeah,
Okay. Let's talk about acquired resistance then.
right.
This is where they learn how to fight back, right?
This is the one where they develop resistance. So these microbes, they were once susceptible to a particular drug, but then through genetic changes, they acquire this resistance after they've been exposed to that antimicrobial.
Okay, so it's after. So exposure kind of like
It's a consequence of exposure.
it forces them to
They adapt.
evolve.
And there's two main ways this happens.
Okay, let's hear them.
First up, you have spontaneous mutations.
Okay.
Now, as bacteria replicate their DNA, sometimes there are these random errors.
It's like typos.
It's like typos, exactly.
Yeah, we all make typos.
Everybody makes typos.
Yeah.
And occasionally, one of these typos happens to create a change that makes the bacterium resistant to a drug.
Lucky typo for them.
Lucky typo for them.
Bad typo for us.
Now, here's where it gets really interesting. When that antibiotic is introduced, it kills off all the bacteria without the mutation. But the one that has that lucky typo, it survives and it multiplies.
So it's like not that the antibiotic causes the mutation,
Exactly.
but it picks for
It selects.
the ones that already have it.
It's like natural selection in action, right?
Oh, that's so cool.
One really key example of this is rifampicin resistance in M. tuberculosis. There's a mutation in a very specific gene, the ARPHOB gene.
Okay.
And this gene, it codes for RNA polymerase, which is the target of rifampicin.
Okay.
So that mutation, it alters that target, and the drug just can't work anymore.
It just doesn't fit right.
It doesn't fit.
Okay, what's the second way they can acquire this resistance?
The second major way is horizontal gene transfer, HGT.
HTT.
Yeah, and this is how resistance can spread really rapidly.
Okay.
It's like bacteria sharing cheat codes.
I like that. Okay, so how do they share these cheat codes?
Well, there's three main ways this happens.
Okay.
One is transformation.
Okay.
A bacterium, it can actually grab free-floating DNA from its environment. Maybe this DNA came from a dead bacterium.
Just floating around.
Yeah, it's just there. And if that piece of DNA happens to have a resistance gene in it, the living bacterium can incorporate it into its own DNA. Just like that.
Just takes it up. It's amazing.
Wow.
The second way is transduction.
Transduction.
This is where bacteriophages, viruses that infect bacteria.
Oh, okay.
They accidentally pick up some bacterial DNA, including those resistance genes, and they inject it into a new bacterium.
So they're like little couriers.
They're like little couriers, yeah.
Oh, that's wild.
Spreading those resistance genes around.
Okay, that's 2 ways. What's the third way they do this information sharing?
The third way is conjugation.
Conjugation.
And this involves direct contact between 2 bacteria. They form this physical bridge, a pilus, and they can transfer genetic material. Often, this genetic material is in the form of plasmids.
Okay.
Plasmids are these small, circular pieces of DNA.
Think of them like a little USB drive.
Like little USB drives, exactly.
Yeah.
That can easily carry and share these resistance genes. And sometimes these plasmids, they don't just carry one resistance gene, they carry multiple.
Oh, wow.
So yeah, it's amazing how they can share this information. So they're either born with it, or they evolve it through these mutations, or they swap this genetic material.
It's incredible.
It's fascinating. Okay, so what about cross-resistance?
Okay, so cross-resistance is when resistance to one antimicrobial agent leads to resistance to others, even if the microbe has never even seen those other drugs before.
Okay, so it's like a domino effect.
It's a domino effect, and this usually happens because either the drugs are targeting the same part of the bacterium,
Okay.
or the resistance mechanism affects multiple drugs in the same class.
So like one change and it's like boom, boom, boom.
Exactly.
Okay, what's a good example of that?
The perfect example is MRSA.
MRSA. Everyone's heard of MRSA.
Methicillin-resistant Staphylococcus aureus. This bug, it produces a modified penicillin-binding protein called PBP2A.
Well.
And this is thanks to a gene called MECA. And this one change, it makes it resistant not only to methicillin, but to most other beta-lactam antibiotics, things like penicillin, cephalosporins, because they all target these penicillin-binding proteins.
So that one change is like huge implications.
Massive implications. One change, multiple drugs ineffective.
Wow, they're efficient. Okay, so that brings us to multi-drug resistance, MDR.
Yeah.
This is where it gets really tricky for
This is where it gets really complicated.
Right.
Yeah, so MDR, or multi-drug resistance, it's defined as resistance to 3 or more different classes of antimicrobial agents.
3 or more.
And these MDR organisms, these are the superbugs that you always hear about.
Superbugs.
And this typically happens when a single microbe, it just accumulates multiple resistance mechanisms.
So it could have one of those natural ones.
It could.
And then it could get a mutation.
And then it could get a plasmid.
And then it could get a plasmid that's got all these other resistances.
Yeah.
Yeah.
And sometimes, the sources point out, sometimes just overproducing those efflux pumps we talked about earlier
All right.
can lead to MDR because they're so good at pumping out a wide range of different drugs.
They're just like, I'm not letting anything in.
Nothing's getting in here.
Yeah.
And so, we see this in these really tough to treat infections caused by things like MDR Pseudomonas aeruginosa, MDR Acinetobacter baumannii,
Okay.
Carbapenem-resistant Enterobacteriaceae, or CRE.
CRE.
Of course, MRSA, we talked about that. Vancomycin-resistant Enterococcus, VRE.
These are the big ones.
These are the big players. And this, it really limits our treatment options. And unfortunately, it often leads to higher mortality rates.
So it's a big deal. I mean, understanding all these different ways that resistance arises really emphasizes why the sources are all talking about how important it is to be careful about how we use antibiotics.
Finding new ways to treat these infections, alternative therapies.
Absolutely.
Yeah.
So basically, what we've learned is microbes have all these different ways
A little bag of tricks.
to
Clever.
resist our drugs. It's kind of like a constant battle between
It's an arms race, right?
us and them.
Yeah, an evolutionary arms race.
So we've got natural, we've got acquired, we've got ways they can share.
They're all connected, right?
It's pretty amazing.
It's wild.
It's a little scary, but it's definitely fascinating to think about.
You got to respect them, right?
All the challenges we face. And so kind of a final thought for everyone to ponder.
Yeah, to chew on.
And we talked about how easily bacteria can share these resistance genes, right, with each other.
Right, through horizontal gene transfer.
So how might this sharing, this interconnectedness,
This web,
actually make this problem even worse?
Oh, it's a big one.
How might this be contributing to a global
problem.
spread of drug resistance?
Something to think about. It's something to think about.
It really is.